The overall goal of this procedure is to quantify permeability of the zebrafish embryonic neuroepithelium. This is accomplished by first injecting a 70 kilodalton fitzy-dextran into the brain ventricles. Next, a time course of bright field and fluorescent images is acquired. Then, the distance of the fluorescent dye from the forebrain hinge point at each time point is measured. Finally, the distance traveled over time is calculated. Ultimately, results can be obtained that show changes in neuroepithelial permeability by comparison of different genetic backgrounds or environmental conditions.
The overall goal of this procedure is to quantify permeability in the zebrafish embryonic neuroepithelium.
To prepare for injection, load a previously prepared injection needle with fluorescent dye. Mount the needle on the micromanipulator and microinjection apparatus. Use forceps to carefully break the microinjection needle to roughly 2 micron in width. Measure the drop size in oil, adjusting the injection time and pressure so that each injection delivers 1 nanoliter. To prepare the embryos, coat 2 dishes with 1% agarose in water for each condition, and using a 200 microliter pipette tip, poke holes into the agarose and remove the plugs. Fill the dishes with embryo medium. Under a stereo microscope, use forceps